So what exactly are histone proteins and why are they essential? So these are basically the proteins that binds to the DNA to condense the DNA inside the nucleus of an eukaryotic cell. Since there is a large material, so we need to condense it somehow to fit it inside the nucleus. Or in simple words, we can say that it supercoils the DNA to fit it inside the nucleus of an eukaryotic cell. Now coming to the discovery part of the histone protein. So histone protein were discovered in 1884 by Albrecht Kozel inside the avian red blood cell nuclei. Generally, the red blood cell nuclei are not nucleated. Red blood cell nuclei and red blood cell are not nucleated, but in avian they are nucleated. And to our surprise, they responded to the stain named DevQuick. The blue stain was observed. Now, to understand how it binds with the DNA, we need to first understand the structure of the histone protein. So, the proteins H1, H3, H4, H2A, and H2B, these are the histone protein who are responsible for the condensation, or we'll say helps in binding of the DNA. So each of the two copies of H2A, H2B, H3 and H4 forms an histone octamer to which our DNA binds. And then there is H1 histone protein that interacts with the linker DNA. What linker DNA is? DNA between the nucleosomes. So this entire structure where the DNA is wrapped around the octamer is known as nucleosome, and the DNA between two such nucleosomes is known as the linker DNA. Now, since we have understood the structure, we are moving to the part where we will understand how the DNA wraps around the histone. So histone proteins are basically positively charged, and the reason behind it is the amino group. And DNA is negatively charged because of the phosphate backbone. So these two interact. And then finally forms the chromatin. Chromatin means DNA plus histone protein. So the histone octamer with DNA coil around it and forms the nucleosome. And the H1 histone protein, it provides the compaction to the structure linking with the entry and exit DNA. As we've already discussed what chromatin is. So if we observe it under the microscope, in the extended or elongated form of chromatin, it looks like beads on the string, where each bead is a nucleosome. And this is how it binds, and this is how an histone protein binds around the DNA. Thank you. It was all from my side.